They should change that voice like once a year at least. Keep it fresh. Good morning, everybody. My name is Jen Yost. I'm a faculty member at Cal Poly in San Luis Obispo. And I also work with Nico here at ASU as part of the Symbiotic Support Hub. And we've put together this conference with the support of Katie Pearson right here and Lindsay Walker and also Jorrit Poelen right up front. And so thank you all for coming. And I guess this workshop came about because I use digital data for my research and I curate a collection of plants. And I've been hearing about this extended specimen for a long time. And I've been frustrated that I can't go to my computer and type in the thing I want and get the results that I'm expecting. And so the goal of this was to bring together some of the front runners who are thinking about these ideas about the digital extended specimen to try to push the conversation forward. And then with a group like you of experts to sort of try to workshop and brainstorm some ideas of what that might actually look like. And so go ahead and go to the next slide. So here's one example of the digital extended specimen. This was a diagram and a paper that came out of a consensus building working group that GBIF hosted. This paper was published in 2022. And the idea is that everything in that blue oval is this digital extended specimen, but there's many questions that remain about this idea. And we've seen so many talks with great diagrams that show all of the linkages and connections. Go ahead. But many questions remain, like, what is this? Where does it live? How do I access it? What's that user interface like? Is it one thing or is it many things? And do we have one concept of the extended digital specimen, or do we have many concepts that fit certain taxonomic groups or disciplines? And then ultimately, what are we doing right now in our daily work, our research, our curation that is contributing, helping, or hurting that ultimate idea of the extended specimen? And so the point of today is to hear from 4 speakers. Go ahead. We'll hear from, you'll see who we're going to hear from. You can read it up there. And then after our speakers to give a presentation sort of about what their sphere of influence is doing related to this topic, we're going to do a group activity together. And as we were planning this, we realized there's just not enough time to get you all to do that workshop and also have the discussion that we want to have. And so we're going to extend our discussion into that 11 to 12 empty space afterwards. That way we can really spend some time tackling some of these questions. So if you are joining us on Zoom, we're going to leave the Zoom room open and also leave the recording open. And many of our activities can actually be done virtually. And then you might want to tune back in for the panel discussion that will probably start at around 11. So to start us off, I'd like to introduce Larry Lannom, who's joining us virtually. He's the Vice President and Director of Information Management Technology at the Corporation for National Research Initiatives here in the US. And that's the home of the handling system and the digital architecture that deals with digital DOIs, digital object identifiers. He's also the convener of a sort of casual and long-running discussion group that has been meeting regularly to discuss using DOIs for biodiversity. And he's the chair of the DISCO Technical Advisory Board and also the co-chair of the technical committee for the FAIR Digital Object Forum. So with that, please welcome Larry.
Hello, everyone. I will share my screen. And someone will tell me if this is working. Can you see it?
Looks great.
Yes, we can.
Okay, all right. So the group that was just referenced is this group. I assume some of those names will be familiar to you guys. And we've been meeting, sorry. We've been meeting since May of 2020. talking about the application of something called digital object architecture, which is a little different from DOIs. DOIs are the an identifier part of digital object architecture, one of several. And applying that to biodiversity data management. This talk will be a very high-level architecture talk. From my perspective, biodiversity is a use case, and it's an interesting use case. And I've been associated with DISCO for a number of years now. We met, I got in touch with Dimitri Kourias through RDA, and that started a series of discussions. This set of activities then converged into the Fair Digital Object Forum. That's taken off recently, and it became clear that our group could leverage that work. And there was a conference, the first conference last October at Naturalis in Leiden, gave us an opportunity to use the DISCO presentation from Sharif, who you'll hear from later, to submit a short summary of our conclusions and intentions to date to a Rio special issue. And there's the title of it there. So that's sort of what we've been doing. Details on what that really consists of coming up in the rest of this slide presentation. The current state is that we conclude this is a good idea. It's very ambitious, as I'll try to make clear, but it's definitely time to move from discussion to implementation. I'll start by looking at this from the perspective of the Fair Digital Object Forum, which is largely inspired, but not entirely consisting of digital object architecture, but digital object architecture is a large part of it. So what is FDOF trying to solve? The general problem is that we've seen enormous increases in computing and network capacities over the last couple of decades, such that data that was formerly just referenced, held in reference from articles and held in labs and so on, is now actually available. And that should make things better. Yeah, we should be able to use all that data to figure more things out and share it and reuse it. This has happened in many areas, but certainly suboptimally. There's overwhelming amounts of data, just very, very large numbers, more than human beings can deal with, really, especially as our systems are oriented to slow humans who will never catch up, never able to put their arms around all of this data. And it's hard to even know where it all is. So opacity makes the common good elusive. Who has the data? What are they doing with it? And the fact that all this data is spread across multiple heterogeneous systems means there's a fair amount of complexity, and complexity always means fragility, which leads to security problems, reliability problems, and so on. So that's the general problem. A lot of data, which should be beneficial, has turned into a problem. Looked at just from the research point of view, you have sort of the same thing, but a research-specific aspect of it, which is fragmentation of systems, lots and lots of APIs, lots and lots of sources, result in a lot of silos, difficulty in moving data from one place to another, difficulty in using data from multiple sources. So interoperability remains elusive. You have a reproducibility crisis. If I have your data and I understand your methods, I should be able to reproduce your results. This has turned out not to be the case. We also, in many areas, we also have funding issues. If I want to reuse your data, you have to publish it. So who pays for that? And then who gets credit for it? You publish your data, I write a great journal article on it, I get promoted, you get nothing. The sum of all of this is that more time is spent on data than should be. And data should be there and easy to use, benefiting science instead of being a problem that has to be dealt with. So FAIR, which most of you have heard of, I assume, findable, accessible, interoperable, reproducible, provides guidelines to address these issues. It's almost too good an acronym. A lot of people interpret it as being, even-handed. It's actually about enabling machine access to things. And it's been applied to a degree to publications, but that's low-hanging fruit. To really take advantage of machine access to data, you'd have to get it back to the data source. You have to implement this at workflows, relationships, and collaborations, and so on, which is a real challenge. In general, we need to turn the challenge, this large challenge of a lot of data and a lot of complexity, into an opportunity. Change the problem of too much hard to use, find, understand data to an advantage, lots of accessible and understandable data. There is also a sort of a misunderstanding that making data available is all you need to do. But if you make data available and you don't make it understandable, you may actually be doing the world a disservice, because it's easy to misinterpret and misunderstand data. If I make my spreadsheet, for example, available to you, and I just have some little cryptic names for the rows and columns, you can guess what my spreadsheet means, but you don't really know enough to leverage it and take advantage of it. All right, what's digital object architecture? Within the FDOF and in general, this is an approach to add a level of abstraction to current information management systems. Now that's not unique to digital object architecture. There's a sort of a old slogan or saying in computer science that every challenge in computing is solved by adding another level of indirection. You just keep abstracting things up. And that's basically the approach of digital object architecture. This is a model here is the internet itself. So why has the internet architecture survived all these decades? One of the reasons it has, basically unchanged, there have been incremental improvements in the internet all along, but the basic architecture has remained the same. It's because it's very simple. It has just two different components, really: an address space, which is IP addresses, and a way to move packets of information from one address to another. That's TCP, with a few others. So that means that the people who engineer the network don't have to worry about what's in the packets. And the people who make use of the information in the packets don't have to worry about how it got to them. This simplicity is the sort of thing we want to apply to information management. We want to create a set of persistently identified and uniformly addressable digital objects. We don't want to have to care what's inside them, how they got created, etc. We just want to be able to name them and know how to use them. For basic information management tasks, every object can be treated the same, regardless of information content. Again, the internet analogy is every packet can be moved across the internet in the same way. It doesn't matter what's inside it. Despite some thoughts originally, we didn't need separate computer networks, separate internets for physicists versus lawyers. They both use the same internet, but the applications at the end are very different. That's sort of the goal of digital object architecture. So it not just applies to biodiversity information, but biodiversity information is a use case from that perspective. Every object has a globally unique and actionable identifier. Every object is typed. You need to know basically what kind of object this is as a head start to how to manage it. Every object has tightly associated metadata. You need to understand about the object in order to use it. Every object has a queryable set of operations that can be requested of it. This is another lesson from computing in general, which is if you think about your data, and if you consider whether this data will be of interest or value to the world 50 years from now, I suspect most of you would say, yes, it is. If I ask you what the what the computing environment will be like 50 years from now, you'll either have to honestly say you have no idea, or you'd have to make something up, which is unlikely to come true. So that means we have to deal with the data at as high a level of abstraction as possible. Give it a name and give it a set of functions. And then how that data is constructed, organized, etc., in the background can change over time. This is sort of what we do with identifiers. DOIs, for example, will remain constant over changes in the ownership and location of items. We want that same sort of indirection to apply to information objects. The information object has an identifier and a set of functions that can be applied to it. And then how those functions are implemented can change over time. And you need then a way to address the objects as well. We have a digital object interface protocol that's available that may, something very much like that will have to be applied over time. Maybe that one, maybe a different one. Why is this a good idea? The digital object model simplifies the solution space. Again, treat every information object the same until you have to differentiate to accomplish your purpose. Push the current cacophony of information management and storage systems down a level of abstraction. If you have 27 different systems, and you build a client to address 27 different systems, you have to understand 27 different systems, and they're going to change over time. What you want is those 27 different systems all to talk to one middle layer, and then your client deals with that layer. And then objects, you want objects to be self-describing. They carry their type and access control information independent of their current system location. That's most easily understood in terms of access control. Right now, most data, the access control information, who can get it, is determined by the interaction of the entity and the system it lives on. And when you move that entity out of that system, the access control information is lost. You want to associate all that with the object itself. This is a diagram that will appear in the paper I referenced earlier. And please note the very bottom line. Existing services continue. So now we're here a little closer, although it's a very high level discussion about network management, network information management on networks. Here we're looking at digital extended specimens, a bit similar to that slide you saw earlier. Existing services continued. We're not proposing to eliminate anything. We're just adding on top of existing stuff and the existing services. So you have collections. And then the bottom layer in the rounded rectangle there, which shows the gears, that's the important part, digital specimens mapping. You take the existing data and system by system, you transform it into, you normalize it in computing terms. You turn it into something that is recognizable across multiple systems. And then above that, you have clouds, which is aggregate the data, virtual collections. And you associate other information with it. That's the extended specimen part. And Sharif will talk more about that in his talk. And then at the top, you have clients. But again, you have the clients only need to understand one interface. They don't need to understand 27 interfaces, as they might have to do if we didn't have this middle layer. So back to our little group. We have yet to write a definitive paper beyond the short article. We need more detail, worked out examples, small trial results, but mainly we need prototypes and funded projects. The FAIR Digital Object Forum is moving into testbed mode our group as a group, or individuals will participate as appropriate. We'll continue to look for funding opportunities and look for collaboration opportunities. And that's the end of my slides.
Kind of nice. Okay, so next up, many of us share data through GBIF, and today we have Tim Robertson from the informatics team, who's also joined by Laura Russell from the engagement team, and they're keen to explore where GBIF can help in our efforts to extend specimen data and answer questions about what GBIF is doing. And so as an introduction, Tim will provide an overview of data flow in GBIF, highlighting the existing tools and introducing ongoing work at expanding the data model. So with that, please welcome Tim.
Good morning, and thank you very much for having me.
Okay.
Can you hear me okay? Yep. So I work for the Global Biodiversity Information Facility in Copenhagen. I'm in the informatics team, so this is going to be informatics oriented, but I'm here with Laura, who works in the engagement team. And we're looking forward to participating in the discussions today. The instructions for our presenters were to try and address some of these points. How will we connect data sources about specimens? What tools do we have now? And what are the limitations that we should attempt to tackle at the moment? So GBIF is, I'm sorry, I just need to move. Okay, GBIF is an open data infrastructure. It's a network of people and institutions. So shown here is a map of where the institutions are located that are publishing data on GBIF. So there's more than 2,050 institutions that share data in GBIF. It's growing at around about 2 per week. For the specimen data that's coming onto the GBIF network, there is a new specimen shared in GBIF every 2 to 3 seconds. So that's the rate of data that's coming onto the network. I'd like to just, from a very high level point of view, talk about how data flows through GBIF so that we all understand how GBIF operates today. And that should set us up for our discussions that follow. On the left, we have the primary sources. So these are the people who are curating specimens and doing research. A lot of that data is held in collection management systems. Those people are preparing, or the systems prepare data to a common data standard. And they produce data sets. And these are registered in the GBIF registry, which is open. And anyone can access the registry and discover the location of these data sets and ingest them. At GBIF, when a data set is registered, or if it's changed, our crawling infrastructure brings that data into a centralized data warehouse. So GBIF are integrating data in a data warehouse system, not dissimilar to, say, IDIC-Bio. As data comes in, we go through formatting and consistency checks, and we do some enrichment. Now, the enrichment that we apply often uses reference catalogs, and we've got APIs for that. And I'm going to show you an example of that because it may be of interest in these discussions. On top of the data warehouse, we've got query services and reporting services, which power our public tools, such as the GBIF website and other portals that are accessing this content. So let's look at an example record and see what happens to it as it goes through this process. So this is a very typical record from the California Academy of Science, an ant record. And I've highlighted 2 things that are a little bit interesting. One is that it's got a link to another specimen, which is shown at the top, and it's got a note that this is a DNA voucher. Now, in California, they run an instance of a data repository. So they've got their own data repository in their institution. It's actually an installation of GBIF repository software. And the data set is that one at the top, which is AntWeb. So by sharing this data set in their repository, it connects with GBIF, and we index the data. So this is what that record looks like in GBIF. I'd just like to highlight that what you see there is the processed view. So that's after it's gone through the GBIF processing. But a lot of people aren't aware that we actually hold the unprocessed views of data. So how did it look as it was actually published from the source system, which can be in a variety of formats. And then that gets normalized against Darwin Core, which is an open data standard, but it's still an unprocessed view of the data. So you can use the GBIF infrastructure to actually access standardized views of data before GBIF have actually interpreted it. As we're processing this data, we use open source software. So that software is available for other people to use. And we also have these reference catalogs that we look things up. There is a huge variety in data that's shared in GBIF. So this is just a fictional example, but we get data with incomplete identifications, misspellings, abbreviations. We have a service which takes that and normalizes it against a reference backbone taxonomy. That's an open service that might be useful for people within their own tools to help enrich or clean up their data. We have similar services for geospatial processing and other interpretations. One of the tools that might be of interest in this discussion is that we have a vocabulary server. A lot of the values contained in our data could or should be standardized vocabularies, standardized terms. So this shows just a screenshot of the life stage vocabulary. Vocabularies are created in lots of communities, but they exist in many different forms. We load them into our vocabulary server that's then able to present our vocabularies in a consistent way for application developers to build tools on top of. We have a few vocabularies being worked on within the TADWIG data standards group. There's a vocabulary underway at the moment relating to paleontological data sets, for example. So coming back to this ant record that we're using as an example, if you come to GBIF and you put in the catalog number at the top left, you will actually find 5 records of data in GBIF. So 2 of them are the specimens that came from California. It's the ant and the DNA voucher. But then, because we index the big DNA databases, we've got the records from IBOL and the INSDC GenBank databases. And thanks to the work of the group PLASI in Europe, we have a record of this specimen being cited in a taxonomic paper. So we start to see that there is more information relating to this specimen in GBIF than was actually coming from the institution. But shouldn't these records all be somewhat related? Well, the answer to that is yes, of course. They're not 5 individual records. If you come back to the view in GBIF, at the top, there's this related records link, and that actually takes you from the specimen to the other records that we've indexed that relate to that specimen. So that's what's shown on the left here. We've got the record from the museum at the top, and then we've got the others below. And the way that we do this is running a clustering algorithm across all of the data. It's basically a nearest neighbor match kind of algorithm. We've explored machine learning approaches to this. The algorithm can definitely be improved, but it's GBIF's attempt to try and detect data that should be connected, and we try and make these links. These are just suggestions. They need to be curated. So people are now beginning to build tools on top of this that then curate the links and say, well, is this actually a correct link? But this could be a source of information that people build systems on top of that actually start to positively connect data together. The little picture at the top right there is really trying to express that, you know, that is the graph of information that happened. The ants were observed, they were collected, they were preserved, they've been cited, they've been subsampled and preserved, they've been sequenced, and they've been imaged at some point. That information model is not clearly expressed today on GBIF. And that leads me to our ongoing research work to explore a new data model for GBIF. And this is the final topic I'd just like to introduce today. We're progressing this work with John Jorrit. He's the lead of the Darwin Core Standard, and it connects with other TADWIG task groups that are underway. So why are we exploring a data model? Well, as GBIF has grown, we've learned that there's a desire from both publishers and consumers of GBIF to handle more complex data. So it's really a demand for GBIF to deal with richer data. We're taking a case study approach to this research. So if you look at the URL shown there, you'll find some of these cases. There's examples for eDNA data, environmental measurements recorded along with biodiversity data, automated recording programs with camera traps, and there's others. We welcome more examples if people would like to come forward with cases they would like us to explore. When we go through a case study, we go through basically three processes. We start with working with a group to try and come up with a clear description of what we are trying to solve here. The way people express their problems are variable, and we try and make a consistent way of describing what's going on. And that's really just a communication exercise of making sure anyone looking at this really understands what we're talking about here. From that case, we then build what's known as a conceptual model, and then we look at publishing models, and we separate out the theory of what's being described here with the publishing models, because publishing models are how we expect people to actually shape their data in ways that we would then be able to extract the concepts from. Conceptual modeling is not for everybody. So I'm not going to go through this in great detail, but if you explore this work, these are the kind of diagrams that you're going to find. And just to give you a little bit of a sense of what's in there. Over on the left, we've got model entities around location and georeferences, geological context for the location, if we're dealing with fossils, for example. In the middle, we've got events, which can form part of a hierarchy of events. So you can have nested event structures following different protocols. There can be measurements and facts associated with events, so environmental measurements that have been captured during the recording, for example. We've got information about physical material, such as specimens, and records that are born digitally, such as media and videos, or genetic sequences, and how they relate, and the chain of evidence that they form. And these, they can be complex relationships between these, and they can be the evidence for why we can assert an organism occurs. Determinations are made against the evidence. That could be against using sequences or the physical material, which links the material to a taxon. It's important to note that in this model, we're able to identify identities representing multiple species, for example. And at the top of this, we have agents. Those are the people involved in their processes and the roles that they have. So this is all very theoretical, right? And GBIF's an infrastructure that's operational. We wanted to test some of this theory. So earlier in the year, we gave some small grants to the groups listed here to try and shape some of their data against this theoretical model. And the overarching idea was to learn, what are the limits of this data model? Do people have data that we can't fit into this structure? We're still digesting the outcomes of this process, and several of the people in the audience were involved. But the kind of early indication is we're not aware of any data that we weren't able to somehow fit into this model. So that suggests that we might be on the right tracks. It was a difficult exercise for many people. So the conceptual model is definitely going to be very hard for a lot of people to deal with. And that reinforces the need for simpler models for how people are actually going to share data. So that's where we are at the moment, is we kind of have this idea that we know that there are people who are dealing with very simple data, and that's what they have the capacity to work with, simple spreadsheets. And at the other end of the spectrum, we've got this very complicated model that's going to satisfy some people, but probably be too hard for many people to achieve. Somewhere on this gradient will be a set of data sharing models, data publishing models that are able to capture the key aspects of people's data and be a new way for us to move data sets around within GBIF. And this is an ongoing piece of research and exercise that we're at the moment. So just with that sort of brief introduction, I hope that gives you a background of where GBIF may play a part as we go into these discussions. But just to summarize, the GBIF APIs may be useful for data cleaning and enrichment during specimen curation. The registry and clustering may help identify links between data as we're looking to extend specimen records, and our ongoing work on the data modeling may help towards some of the structure around the extended specimens that we envisage. Looking forward to the discussions, and that should say thank you. Thank you very, very much.
Okay, next up we have Jorrit Poelen, who is an independent software and data engineer. He's a Ronan Scholar and is affiliated with the UC Santa Barbara Cheadle Center for Biodiversity and Ecological Restoration. He contributes to Global Biotic Interactions Project and the Terrestrial Parasite Tracker, the Big B TCN, and the Open Traits Network. And I am supposed to inform you that he will work for coffee and cookies. So please welcome Jorrit.
Thank you, Jen. Yeah, thanks for having me. And I'd like to share some of my experience in working with the Open Traits Network, and also in helping to start and create global biotic interactions. And yeah, so it's a use case, and an alternate title is on keeping verifiable digital knowledge available through collaboration, trust, and mutual benefit. Right? Because what I've learned over the years is that at the heart of everything is just that one person that curates the data set, right? One of the many data sets that we all rely on and try to use as a community. So Jen asked me, like, what does a decentralized extended specimen look like? And then what kind of roadblocks did he, being me, encounter building this resource? Yeah, what's needed? So yeah, so I had to answer these questions. And so I realized that Jen actually introduced a new concept here, a decentralized extended specimen, which was, something I had to then define. So I hereby announce the release of the first version of this decentralized extended specimen. I call it the Yoast Decentralized Extended Specimen, also called YODESpec version 0.1. I try to make it sound very official because it is, right? So here's the definition. It's like a long definition. An ever-evolving collection of particular verifiable versions of subjective digital compilations of existing versions, verifiable digital traces associated with a physical specimen. Should I back up, or are you still with me? As published by a group of trusted collaborators, enabled through mutual benefits. So this to me is it may sound like a joke, but this is really what I believe in. Yeah. So what does that look like for me, right? Because it's a very subjective thing that I work on with a group of trusted collaborators for those cookies and coffee, right? I need also peanut butter sandwiches because cookies are not enough. So looking at Globi, so I started to work on this about a decade ago with the help of many others. It's an open source, open data, collaborative thing. It's very tool oriented because my background is software engineering, so I'm I think as a software engineer, right? So I thought, oh, this is an engineering issue. But it turned out, you know, not to be that. So in a nutshell, you can register data sets about species interactions in any format. Doesn't have to be standardized as long as it's open and digital. And then Globy sort of figures out a way to automatically track and index and review those data sets. So it's sort of like a review service. So it's sort of turned from an aggregator into sort of like a data review service over the years. And then users, or humans, can discover those data sets and have another perspective, maybe of their own data sets as seen from the eyes of someone else, like me, a naive view. So here's an example of such a collaboration, actually, trusted collaboration. It took years to develop this. It actually started with a lunch conversations at the Digital Data Conference at UC Berkeley many years ago, 2016, 17. Yeah. And that's where the trust building started. And once trust was built, the magic started to happen. So I learned that Arctos sort of works in a particular way, and I tried to make sure to write tools to adopt to that. Here's a sort of a rough sort of overview of the workflow that Globy has. It's roughly like version your data, right? Just like you sample nature, you also sample the digital world around you, and then align a bunch of names. So this is where the headaches start, right? And then also extract interactions and then somehow sort of mush it together. And then you have some interpreted interactions that can be exposed in many different ways, like giant TSV files, search index, et cetera, et cetera. So the most important thing I've learned is that to keep the original data, because the input, actually, this stuff, it turns out it's not sticking around as you might expect. I've seen projects come and go, and giant, very valuable data sets, they just disappear overnight. No one noticed, right? So I felt sort of like more of an urge to invest more in tracking data sets. So the talk I gave yesterday is sort of a result of that. So looking at a very specific example here of Arctos. So they have a, so Arctos is sort of like a co-op or collection of collections. And one of them is the Museum of Southwestern Biology. They have a very nicely linked data set, which is the MSB Para data set. And they link to all sorts of other collections. And they link with very sort of quote unquote deep links. So they linked from specimen to specimen, which is kind of cool. I thought, but it's kind of a headache to actually resolve those links and connect it. So that's what I that was a challenge, and that's what I took on. So like a couple of examples of collections here that they integrate with. MSB host collection, so that's in-house, but also with folks at Harvard and the Smithsonian, and many more, right? So just an example of what happens if you start linking stuff. So seeing things from different perspective, right? Looking at the same data from different perspective. So this is the Arctos perspective of a parasite record. It sort of looks like this. First time I saw this, like many years ago, I thought, oh, these people are stuck in the 90s. What's going on here, right? But then I sort of realized that it's actually exactly what they need. And I learned to sort of, understand that different groups speak their own language and use their own technology. And this works great for them. It took them many years to develop this. And notice that they are using some kind of a GUID thing, some kind of reference that also sort of looks like a URL. And then there's all this sort of very rich data, and it's linked, right? So the the Eric P. Hauberg, that's actually a link to information of the person that identified it. So again, it doesn't look fancy, but it's like it's all there, right? And then here's the collected with and parasite of, and the parasite of is the one that jumps to another collection, right? So the host is somewhere else. So now this is the way that Globy sort of looks at this data, right? So there's like two things that sort of interact with each other, and then they have a name, right? And if you just look at the Arctos stuff, then you have this sort of rich data here, but then over here you have a link to another record, but the name is not known, right? Because you have to click on the link, you have to follow it. So I made a little robot that clicks on the link and gets the data. And I'll get to that later. If you go to this, if you click on the link yourself, then you get to this landing page. You get to see a name, right? So that's Nomenescio is no longer Nomenescio, right? It's a name. And then you can start to do stuff with it. From their perspective, the parasites is like, yeah, there's a parasite. We don't really care, but but Arctos knows about it. So if you combine the two, you get a real neat view. Neither of them have that view. But now Globy, Arctos links to Globy directly, has that view, right? And it's linked, to the data sets, et cetera, and it's also linked to all these other things. So these names have references like in all sorts of different places, right? So now we have linked or extended or whatever you want to call it, this specimen from Globy's perspective, right? Yeah, and then this was a year ago. And if you look at the difference between the two, you might notice like a teeny tiny little difference, which is the Scolia link here, which is actually something I would encourage you to check out. It's sort of something built on top of Wikidata, and it offers like a really cool view into all the research done on this species. And it came about through a connection I made on the Open Trades network. Linking all the data, it's like a messy family of, like a dysfunctional family of like passionate people, right? And the Open Trades network actually had a similar view on this. So they recognized that this is our research community, right? Where the nodes are people that collaborate and the resources. And then it's easy to think of like integration as this, where everybody sort of hangs out at this one place, this one center, right? And then that's where the magic happens. But in reality, we all know it's more like a decentralized but connected community of people. It looks like a mess, but actually, if you look at under the hood, there's a lot of great stuff happening. So again, switching to Globy's perspective on this, it's a continuous progress or process. It involves all sorts of different people. And then in a recent publication of from the Open Trades Network, they made a way better figure about this process, right? It's a continuous process where you continuously sort of discover, collect, integrate, validate, analyze, publish, right? So everyone is doing this. So there's alignment in the way we think about integration. So it's a process, it's not a product in my mind. So what roadblocks did he encounter, he being me, is time. And so the answer to the first question, the first roadblock is time, which is, it takes time to do this. And then the second one, what to do to make it better, is time and elbow grease. So just keep at it. So now I'm wondering, what does the version 0.2 of the decentralized extended specimen look like to you, right? Like what roadblocks did you encounter? Do you think someone else is going to solve the problem, or are you going to work with your collaborators and get a little bit better at linking across projects, either through email or post-it notes or fancy infrastructures, right? Integration comes in many different flavors, in my mind. So there's a bunch of partners for Globi. This is not a full list. Actually, this is from an old slide. Big B is not on here yet. So sorry about that, Katja. And there's just so many people involved in projects like these. All the open source developers, thousands of researchers that have toiled, have spent many hours in their collections, et cetera, et cetera. So it's like, it's hard to keep track of everyone. And also many different groups have supported these kinds of collaborative efforts. And yeah, so it was a quite a pile of cookies to eat through, and hope there's going to be more. So thank you.
Thank you, Jorrit. Next up is Sharif Islam. He is the data architect for Disco, where he's responsible for overseeing the data and system integration. He participates in the European discussions concerning fair digital objects, and he collaborates closely with a team of developers around implementing persistent identifier infrastructure, digital specimens, and various related services for Disco. So please welcome Sharif.
Thank you for inviting me and organizing this. I'm really excited to talk about what we're doing with Disco and how it fits into the global discussions. And while I talk, you'll see some of the problems and roadblocks we're dealing with and the helps we need. So yeah, really excited to talk about this. So a monkey, a giraffe, a dolphin walk into a bar. Okay, you know the situation. So the bartender says, "Wow, what an eclectic group. Are you all compatible?" The monkey replies, "Well, we're trying to harmonize our data, but it's like a jungle out there." The giraffe nods and adds, "Yes, reaching for interoperability can be quite a stretch." And the dolphin chimes in, "And swimming through different systems can make you feel like you're in deep water." Sorry, that's my dolphin. And the bartender laughs and says, "Sounds like you could use a data translator. Let's raise a glass to seamless integration." So I didn't write this joke, but it's actually quite captures our current situation, and we do actually have a data translator that we deal with things. I'll let you guys choose which animal you are identifying with. But while I was thinking about it, I was also thinking about the history of our community and some of this data, and you've already heard some of this. So if we kind of simplify it in this, simple 3-act structure, I know like it's probably more complicated, but let's say, we have the setup in the Act 1, right? So we've done a lot of work with Darwin Core standards, ABCD, both, on the geological and biological side. We have amazing community in Tadwick and Spinach. We have infrastructure with GBIF and IDIC Bio and others. There's lots of things happening with, digitization and data mobilization, but then you get to that, climax where you're feeling like, things are going to fall down, right? We have more and more data, we have lack of time, expertise, and resources. We're seeing linking issues, interoperability issues, enforcement of some of these policies, semantics. What do you mean by these things? We talked about it earlier as well. More and more, we are starting talking about, AI and machine learning. How do you trust, reuse, provenance, versioning? There's human in the loop for all of this automation. So there's a lot of things happening. Of course, there's solutions for these things, and we're trying to. But you feel like, it's something's going to happen. So what's Act 3? So that's where kind of what we're discussing now. So it's really great that we're here. It could be something hybrid and interoperable. It could be centralized, decentralized. We don't know. There's several ways, as Jorrit just mentioned, way to think about these things. But one thing for sure is the FAIR and the digital extended specimen ecosystem should be there. So I will briefly introduce Disco and the idea of the digital specimen and some of the stuff that Larry talked about from the digital object side. We will see how a little bit concrete this is, and a few examples of this data harmonization and what we're trying to do now and some future approach. Disco is a research infrastructure in the making for the natural science collection. So one thing that worked out in Europe is that there were already several other research infrastructures in biomedical domain, in digital humanities, in astronomy that kind of helped us to make our case that we need this for collection holding institutes. So these are natural history museums, herbariums, and small collections all over Europe. So we're working with 23 countries with 170 museums. So the idea is that we need a coordinated effort to build a research infrastructure that will not just digitize, but provide service, training, and capacity building as a sustainable effort. So that's sort of the funding and the structure. And the technology side, we started looking into existing systems and then slowly moved into this idea of digital specimen and fair digital object. And then the whole extended specimen network came in. Basically, the idea is that the digital specimen will be sort of the central element in this architecture as a digital twin. So not just a record, there are other things happening. And I will give you an example. Counterpart of the physical specimen in a collection. And that sort of builds in the whole service ecosystem. We just finished our preparation project. You can find a lot of information on the website, how we propose some of the things and some of the prototype we're building. And we're excited about the next phase where we're thinking about operationalizing some of the services soon. And again, these diagrams, but to identify a few things from it. So we're working on something called the Opal Digital Specimen Specification. And this is where some of the Data Harmonization Act will come in, where, Darwin Core, ABCD, and others, we're trying to harmonize them. But then we get into sort of the digital specimen and digital object architecture specific. We need to identify precisely and unambiguously the digital specimen. So DOI and other identifiers are perfect for that. Even though we have the humans in the specific interface, a lot of these things are targeted for machine actionability and machine readability. So if you give the machine 10.22/ that string with the metadata, it can figure out what it is with the structured, and then do processing. But we need that description. We need that semantics and attributes. So that's why these three aspects, I think, for building the infrastructure is important. We need the identifier. We need a specific way to describe it. And of course, the operation and the use linking and other things are also important. So just to give an example, where will Disco fit in? We heard about Globi, we heard about our content management systems and other infrastructures. So currently, we have all these different systems, that Larry also talked about 27 different things, but we also have the aggregators on the other side. We are working with GBIF and also the geological specimen as well. So in a way, you can think of Disco as that middle layer, that object layer. This does not mean that you cannot talk to GBIF and others directly, but when you have that middle layer, you provide that harmonization and identifiers that will come through for other uses. And this is just one interaction. You could have, community curation. You could have other services that build on top of it. And the big idea we have is that we can have these components shareable either with the content management system or in the aggregators. And more and more, we are going towards automation and machine operation. So of course, the human users are important because those are the ones that will take care of the data. We trust them. But if we are dealing with millions of records and we want to do something better, quicker, and then spend time on doing the exciting science, we need to kind of make sure the machines can take care of it properly. So in a way, this is where we see Disco. Of course, this is evolving. We're talking with all of these people, from both sides. But this idea of the fair digital object and digital specimen is kind of what we think that middle layer will connect some of these infrastructures. And these are another view of how we see different services coming along. Some of these things are operational, some are in the prototype space. For example, we're looking into how researchers visit and ask for loans across Europe. It hasn't been happened in a coordinated manner, so we had a grant for that to build this European loan and visit system. At the moment, the visit part is working. We're still, working on the loan part. Basically, you can coordinate some of this visit activity throughout the institution, but you can also have the tracking of those things when you publish your findings based on those visits, and then you can get put the credit to the institutions. And of course, behind the scene, you have the object repository and identifier infrastructure processing authentication, API, and all those things. So yeah, why do we need the data harmonization? We need that persistent link. So we need to be sure that when we're talking about this digital specimen, this is the one, and that's the physical counterpart. You can still have your local identifiers, local practices. Behind the scene, you can have 27 different systems, doesn't matter. But when we're linking to that specimen in the extended specimen network, we need that link, unambiguous, machine-readable, persistent link. More and more, if we want to do batch processing, we need to be clear about which data centers we're using when we're harmonizing, what terms you're using, what's the value. So that's where harmonization comes in. And we need different levels of abstraction, as Larry mentioned. We will have some common metadata. As I said, we're dealing with botanical specimen, paleontological specimen, geological specimens. We need some common metadata to enforce, but will be optional extended strategies. So there are levels of metadata that we have to deal with. And as you all know, there's multiple standards and collection management systems, different specimen. Even within a single data standards, we see a lot of variation how people are using it. You might be aware of some of the TEDWIG activity, specifically called MIDS. So this is coming specifically from the digital specimen idea, but also digitalization pipeline. So you might know that when you digitize things in a large scale, sometimes it takes weeks or months for those data to actually be available out there for to be useful. So one idea we have that we can have a minimum metadata about a specimen in a digital form available on day one. It could be as simple as a persistent identifier and a scientific name and the location of the museum. That's it. So that could be what we're calling MIDS 0. You get it out there, you can enrich it, and then the external data, extended data can come in as you take images, as you put sounds and video recordings and things like that. So that will be much easier for you to link that to publication, to GenBank, to GBIF, and other things. And some of these terms are not in Darwin Core or ABCD that we need, but we're not creating a new standard. We're trying to specify it within the digital specimen universe, and then sort of use as much as possible from existing standards. We're also thinking about the provenance, specifically the digital provenance. Who created these records, who annotated it, and of course we also need fields to not just link but describe the relationship. Similar to the Biotic interactions, we also want the digital specimens and other digital objects to be interacted with each other. So if a digital specimen is cited in different publications, how they're cited, how they're used could be an interesting description of the relationship as well. Don't worry, I'm not going to talk about taxonomy. I'm not a taxonomist, but when we started looking into different data sets coming from different organizations, we're seeing all of this, the key value pairs or all of these elements. And this is just looking at the key part. When you look at the value, actual content of the data, we also see a lot of different things. So this is just the field of name or scientific name. And people are using it, you can see from Dublin Core, from Darwin Core, different ways of thinking about it. And within MIDS, you can follow the GitHub discussions where, we're discussing to think about how we have this mapping. We cannot enforce, down the pipeline to change everyone to one name, but you can have some of the mapping when it comes to the digital specimen universe to enforce or harmonize these things. So here's another example. You have Darwin Core scientific name, your typical, binomial, but then you also have ABCD, which has a little bit more structured hierarchy, and then we have the geological specimen where it might just say rocks. That's it. I mean, that's their way of describing things. But we want to bring all of this together within the digital specimen one, and these are real data that we have seen when we're ingesting. Yeah, this is also another way of sort of thinking about the flowchart, and we have done a lot of documentation in the GitHub. I suggest you quickly look. Basically, when we ingest and we're going line by line, for the specimen record in the Darwin Core archive, if we find a mapping with Darwin Core ABCD, we say, okay, nice, take it. If not, we try to figure out if there's another mapping, or if needed, then we need to discuss for a new term. So some of these terms that we proposed came from this kind of activities. So all of these things also needs some user interface. So this is what we have right now. You can go to sandbox.disco.tech. to test it out. So the search interface is very nice, but it's nothing new. You're probably familiar with some of this filtering, but behind the scenes, there's a lot of things going on. Of course, you can see all of these boxes where we have these sub-disciplines that went through a lot of discussions, how to combine them, because some of this data does not necessarily come from the provider. So we have to also think about how to nicely organize that. But I'll quickly mention some of the PID and metadata stuff that will be important. So this is a very simple specimen record. At the moment, it's very similar to what you can find in the museum portal or in GBIF and other places. But there are a few new things that this will help to get to the digital extended specimen world. The PID on top, that 20.500. So at the moment, we're using the handle system, which you can use DOI. You can resolve it with the DOI as well. So if you put doi.org/this string, it will resolve to the content of this specimen record. And there's some very minimal metadata, and then we also automatically trying to calculate the MIDS level, which in the Tadwig page you can see what that means. That can also automate some of the digitization report and processing. So talking about the abstractions, the one thing we did is that this idea of fair digital object, as Larry was talking about. So there are, if you go back, you see some of this very specific scientific metadata that are tied to the specimen. So this might be very different for a fossil, for a mineral, or even, let's say, archival object. So we want to have some commonality for all of this metadata in that second level. So we have the persistent identifiers, which when we ingest, we create a digital specimen, we assign PID to all of these records, but then we also add these extra metadata. So for example, digital object type, we can say it's a specimen, we can say it's an image, and then all of these things could have optional attributes. For example, if it's a fossil, we can have extra things. And then if you want the actual record, the full record or the full image, then you go to the actual repository. It could be GBIF, it could be the museum, or it could be other research infrastructure, and there's operations and services attached to that. So technologically, what's different is that second layer sits in the global handle server. So it's separate from what you're maintaining in your museum in other infrastructure. So imagine the third layer goes away, or third layer gets redesigned to something else. I can still go to the PID. I can still go to that middle layer of metadata. I can figure out what this object is, the history of it, and the provenance of it. And then ideally through data curation and policy, it should point me to the new location. So that's usually how we do the PID work, but this second layer is something new that we are proposing in the fair digital object world. And of course, it's just a technological implementation. There's policy involved, training involved. So it's not just adding the PID and the metadata would not magically work. We also need to work on the implementation and policy and governance as well. So this is what we call a PID FDO record, which is basically that middle layer. So this is one of the DISCO FDO records, and some of the 100 index gets automatically added by the handle system. But in the middle one that I highlighted from 1 to 44, it's what basically you can think of it as the common metadata that could be, for at least within DISCO, that could be applicable to all objects. So we describe the type, the license, who created the PID, where's the DOI and things like that. And then when you look at the 200 and later, you see some of the very specific specimen related things that are there. So again, this basically sits in the second layer. When the PID is created, we attach this extra metadata. And then, if you want more, you resolve this with the handle.net or doi.org/the string, and then it takes you to the actual record, whatever that record is. So this helps you to sort of set up some of your automation. So for example, you want to get only digital specimen from, some natural history museum in Sweden. Just tell the system. So you have to build the interface to talk with that. Tell the system, just give me that. So there will be millions of PID that coming in, and then you can decide, okay, I want to do this kind of process. I want to do only MIDS level 3 process that has images. So all of this metadata could be, inserted in different level to operate your operation. Okay. And as you can see, there are other PIDs here. We have the ROR and ORCID ID also as well. And this is some of the detailed JSON record that you can see, and some of the new things that we're adding is, of course, the handle ID, the MIDS level, and the version of the specimen. The PID will always get you to the latest version, but you can also go to the other version if needed. Yeah, so some of the work we're doing now, we want to add annotation, automation, and some batch processes with that. We also need help with testing some of these things, specifically for identification. And also interested in sort of training, what does mean for data curation, stewardship for that. And if we have more test cases, we can try it out. We're also experimenting with the RFC process about some of these aspects. For example, what goes here, what we should call it. So we also want the community to be involved in that. And there's a lot of discussions about terms and vocabularies. So we have a lot of things in the GitHub. So I suggest check it out and add questions and be part of the discussion. So yeah, I want to thank, of course, our funders, and some of my colleagues are listed here. They worked a lot to the current version we have in the sandbox.disco.tech. You can check it out. And thanks again for this opportunity.
Okay, thank you all very much. And so now at this point in the presentation, we've heard 4 different talks from different perspectives. And we also on our panel today are joined by 3 additional people. We're joined by Conrad Schock, Jutta Buschbaum, and also Laura Russell. And so all of us are going to have a discussion. We'll answer some of the questions that have come in. We'll open it up to all of you who might have a perspective to add to this, and also questions of your own, and we'll have a group discussion. But first, we're going to experience the challenge of extending the digital specimen. And so what we have scattered throughout the room are big pieces of white paper and some markers. And so we'd like everyone to huddle around the nearest large poster. And this is going to be your group for this exercise. Go ahead and go to the next slide. So gather around those, introduce yourself to the people in your group, and then I'll explain what it is that we're about to do. And there is one right up front with maybe only one person at it. And if you would like to just join, I would provide your perspective on this. I think we're also what people are for Poelen, yeah. What's happening? Yes, yeah.
Oh, right.
Yeah.
I don't have
Just grab a random group. You're getting to know your groups. You have 2 specimens at your piece of paper, and your job is to pick one of them to sort of workshop with. Now, if you feel like, oh, and look away from the front. If your group is very large, feel free to split those specimens into 2, and you can workshop it however you would like. Go ahead and go to the next slide. So these are the 6 specimens. And what we're hoping that you do is you put one of those specimens in the middle of that large piece of paper, and then you're going to create linkages that you actually care about, not all possible linkages, but you, as the scientist that you are, create the extensions of that specimen that you imagine are useful and valuable, however you would like. And then you have 2 different colored markers there, and you can use those 2 different colors to indicate linkages or data that currently exist, and that you know that those linkages or data are there, or those that don't exist at all and you wish that they were there. Okay, so first draw with markers, and we'll give everybody about 10 minutes to do that. What was that? Great, we'll bring markers to your group. And if you haven't already introduced yourselves, sort of giving people the context for where you're coming at, maybe you work with digital extended specimens all the time. Please do so in your groups. Okay, 10 minutes, go. Just a little bit, you could probably do that all day. Let's hear about some of the things that people wanted to know about that specimen. What were some of your bubbles? And feel free to hold up your big piece of paper if you would like. Who's got a data source they would like to connect to that specimen? Gil. But you guys wrote a good one. I can see it.
Conrad, you want to start? Okay, Conrad and I are going to do this one.
Not just stand up so people can see.
Really? Okay.
Well, just you know.
So we put the TC fly in the middle and decided to focus on sequence data. From sequences, those sequences can then be used to confirm species determinations in environmental DNA data sets. And those data sets can be published. So multiple publications could occur from the museum specimens and from the published data sets themselves. And I'm going to pass it to Conrad.
Yeah, and we also had a TBA section of unknowns that could be linked at a later time. So we want to be able to enable that to happen. An important part of this is also verifying for accuracy. So you need things like type material sequences in order to make the links real.
And on top of that, though, that because those sequences were used in the environmental DNA data set, then that specimen becomes an environmental type. So the link goes back to the specimen. So it's really, I feel like a lot of ways, combinations of these data sets that are published, linking back to the original material and to the sequences.
Right. How many of you had sequence data as a component of your extended specimen? Okay, about half the room. Who else has another data type that they would like to connect to their specimen? Go ahead, Tim. Tim.
I'm sorry that you're listening to me a lot today. So our group was perhaps influenced a bit by the fact that I'm in it, and Teresa was in it, and she's been involved in this modeling work. We picked the fish, the preserved fish from KU tissue database. So in there we had the material for the tissue, which was related to sequence data, and it was linked to the source fish that the tissue was taken from. It was clustered, and that information was available. It was also, we saw in the GBIF system, it clustered with another data set for how that fish was actually collected, which was a USGS survey bottom trawl protocol data, bottom trawl data set. So we were able to link a bit of data about how this fish was actually collected from somewhere outside of the museum. And we could link it to the collection and institutions. The kind of things that we've got on our sheet that were perhaps missing was the sampling protocols for how the sequencing was done, and the sequencing protocol, environmental data that was associated with how the fish was collected. We're fairly sure that would have probably been in somewhere in source databases.
How many of you, just to pause you right there, how many of you had something about the climate or environmental data about where the organism was? Okay, so nearly everyone. Okay, fascinating.
We did, this was a quick exercise, but we didn't see the collection protocol. We knew it was a bottom trawl, but that was just the title of the data set. We didn't see other specimens that were collected in that trawl, but they must be linked somehow. The people involved in all of this, I know that they're there somewhere, but they're probably not represented as well as they should be. We've got images, traits, measurements, and possibly other identifications on the material.
Yeah, how many people had some measurement of the organism as a component of their data? Okay, just a couple groups there. That's very interesting. Okay, we're going to move to the next part of this activity, which is that what we'd like you to do then is to pick one of those linkages, and we're going to digest it a little bit more. Can you pull up the Google Sheet? Or actually, it might just be the next slide. Keep going. Okay, so a couple points of what we have. There's 3 main links that we're going to work with today. So one, I wanted to keep you analog for a little bit, but now we'll go digital. So we have a workshop idea board where we have a list of data resources, and then we have a column there for challenges, which might be the next slide if you want to just click over to that. Nope. The idea board. Anyway, it's one of these. Okay, so here's what the idea board looks like. The Zoomers have already been working at it. So challenges encountered while doing this next bit, solutions that you might envision for the challenges that you're about to encounter, and then action items that you think are essential. So this is operating, and we're gonna use this to help frame the discussion afterwards. And then the other link is to this presentation. So we would like somebody who's technologically advanced to snap a photo of your bubble map and then drop it in your in this presentation so that we can all see each other's bubble maps. And then the thing that we're going to work with right now is a Google Sheet, and that link is on the wiki page, and that spreadsheet looks like. Yeah. But will you actually just jump into the presentation, because there's a QR code too. Here is the QR code for this next part of the activity. So what we'd like is at least one member of your group to go to this sheet, and you're gonna put your group members' names. You're gonna add, you can make up a group name, and then whatever specimen you're gonna be extending today. So the 6 specimens that are out, there's a tab on the spreadsheet with the URL to the actual specimen link for them. And so you can just copy and paste that in right here. And then we'd like you to write a sentence describing which specific linkage you are going to attempt to accomplish with your specimen. So what are you trying to do in English? And then you're going to add those URLs as you scour the internet. And this is the elbow grease and time component of what specimen is being extended, where did you find it, what resource is that you're using, and then some type of identifier. And if you go one more, there's an example tab. And so this is one Lindsay really fleshed out, and she gave you lots of examples. So you could have a specimen occurrence in GBIF, but then you have an original taxon description, for example, and that is cited in a thing. So we're going to put these connector words on whatever linkage you're trying to create. So just pick one of them, and you might only have one row right here. But in the example tab, we're giving you lots of.
It's amazing to see that Lindsay was able to do this. I'm like,
give or take.
2 minutes, that's all it takes. It's great. Okay, so is it clear what to do now? Somebody in your group pull up the spreadsheet, and there is what I just realized is that none of you know what your group numbers are. So just claim a group number, and if somebody else is typing in there, get out of it and go to a different tab. And so that's going to be a minor complication there. Okay, so we'll give you 10 minutes to actually get on your computers and attempt to link these things. We can see on the board how the various groups are doing. So we're back to here. So we have things like, let's just go. Team Lightning. Team, whoa, that's a lot. Did you just copy? Did you just copy Lindsay's? It looks very similar.
We're editing it down.
Okay. Also, let's see, Team Whale. Okay, so Team Whale has found some links to 3D scans of that specimen. That's cool. And it looks like also a pub from the Royal Society there. Let's see what group 6 is doing, working on it. Okay, group 2 found some specimens from the same parv order found at the same site. And this is, we're looking at the fossil, okay. Team Canary is looking at a wood specimen collected from the herbarium specimen. Okay, and the specimen said it was collected, but we didn't know where it was deposited or if it was available. Okay, cool challenge. Here is the citation to the occurrence data set. We're trying to find literature linked to the specimen. Could you guys find any?
No, having trouble.
Having trouble, okay. Very interesting. Fish and chips. Oh, fish and chips did a lot. Let's see, they found this is the fish specimen, that there is muscle tissue, and there were 5 other specimens collected together at the same time. And then there's a couple different sequence records. Okay, that's cool. And then some event metadata. Very cool. Okay, Team Iguana. We've got a trap density network. Okay, and this is the tsetse fly. We've got some publications, some field notes. And this is all hypothetical. Because I was impressed for a second. I didn't look at that column. I was like, wow, the extended specimen is here, everybody. Okay, that's great. And let's see what else we have going on. We have species distribution. Okay, so the ranges of those various, this one is the fish. Trying to figure out the conservation status of that specimen. Okay, that's great. And then we've got, let's see, this is the tsetse fly. And it looks like there's some, okay, good taxonomy from GenBank and also some pubs associated with that. Let's look at Team Fly. Okay, Team Fly is also populating some links where we have the record in GBIF. We've got the record from the original collection. And if we look at Team Fish tissue collection in the web portal. Okay, and that's searching within KU's online database. Okay, so that's fantastic. And I want to encourage everybody. So that was, you were all there trying to attempt to do what we all want some service or some tool to do, which is to crawl through the internet and find these linkages. And then from the Zoomiverse here, Deb Paul put a link to this, which is a Wikidata thing, and said that some students have been putting together these web maps, which is pretty cool. You can click on them and see the connections. So this is like a more dynamic version of what we did on the paper there. And this is sort of what I want, me personally, when I think of the extended specimen. I want to extract all the data from any one of these hubs, but this is a cool visualization and the connections between various specimens. That's cool. If you haven't seen that before, that's in the Zoom chat, and we can put this link also in our conference materials. So the last activity that we would like you to drop into your Google slide is on that piece of paper that you have a blank piece of paper on in your group, is to actually draw what you would like to do. Like, imagine you're a software engineer and you know how to do everything. just draw what you want to do at your computer. You go to a website and what do you enter? Like you actually, as a researcher in your own work, what is it that you would most likely enter there? And then, you know, what is that system going to deliver to you? So your inputs and then your outputs, and sort of visually what you would like that to look like. And this is a very rough sketch. So we'll just take 5 minutes to sort of brainstorm what you think that might look like online. Okay, 5 minutes.
And Debbie Paul wants to clarify that it was Shaban who shared this link with that she got from Sharif. So, you know, extended. The slides will be in also on the wiki. The link is on the wiki. And I'll bring that back up as well.
to report out? Like, what's your imagined user interface? Who wants to share? All right.
All right, so something super duper simple and user friendly that you would not need to be a museum person to use. That would basically have two input boxes, right? One that would be the name of the specimen that you want to look up, and it could be common name or scientific name, and also correct for spelling and synonyms like a Google search could. And also another box that would have optional filters to add more boxes with Boolean operators, you know, the and, or, but categories, but that you could type in any keyword. So plant phenology, or the name of a location, or the collector's name, really anything that was just open keyword friendly that you could make more complicated by adding more rows. And the output would hopefully give us one of those beautiful, like, movable knowledge graphs or webs that you were showing. Other specimens of the same species, if you indicated one species you wanted to look up, other locations, if you had indicated phenology, some like color-coded graphs, just everything, link everything. So that was our idea, kind of keep it simple, but have the output be really extensive.
All right, who else has a vision?
I guess I like the idea of kind of attacking it from multiple points. So if you think about like starting with a search, you may be searching on taxonomy or geography and having a system so that it's going to take into account like the level of the search. So you might have, if you're searching on a species, you might have a link to the original species description. But then as you start digging into an individual specimen, that it would kind of branch out to different things. So kind of more like generalized extensions, and then specific extensions to an individual specimen.
Right. Anybody else with a vision they'd like to share? Gotcha.
I like the idea of a spatial search where you can draw or input a polygon, and I think this actually has existed. Maybe Data One has something similar for searching their data sets. You can draw a polygon and then retrieve all of the data sets or all of the linked data that is associated with that polygon.
So we were discussing in our group so we'll explain a little one part, and then the second part, let me okay, the second part would be depending on what we want, like to prioritize what the results are, and the first part, or
Yeah, so I just kind of basically let me select what field I want with the ability to kind of do interoperable things, and then just give me lists of columns to deal with. I don't want analysis. I just want to kind of see what's there. If I want analysis, I'm going to export it later and deal with it. Just like give me the ability to kind of look at the data, quickly sort it, and then like do with it what I want afterwards. Just a really simple reduced state, and kind of keep it uncomplicated and easier to deal with.
So everybody likes the pretty things, the graphs, and those are nice. But anybody who wants to work with the data wants it in a spreadsheet. And those two things are like opposite ends of some spectrum. And I don't know how to get people to actually get on board with the idea that that's hard.
Your vote is just the spreadsheet. That's all you want.
No.
You want both?
That's what people want. That's what people want. You just said that.
Yeah.
I asked for it.
I will go do stuff with that
Yeah,
data.
exactly.
It would be good to ask the people who've proposed kind of search interfaces. Do you expect them to work in a browser, or would you want to be able to search from some other piece of software?
How many of you have the expectation that you would be searching within a browser versus a piece of software? So browsers, there's the hands for the browser people. How many people imagine sort of a standalone installation of software?
Jutta says both. Jutta makes the point that if you don't have stable internet, you don't have internet connectivity, then you will not be able to access something from a browser.
I'll just explain why I made that point. Is just that I observe when I go and talk to users and help them out with access to resources and stuff. They've got like 50 tabs open on their browser, and there might be better ways if we're thinking really creatively about user interfaces how we could deliver stuff.
There's no one in this room who's ever had 50 tabs open on their browser.
I just think it's also important to consider that what works or what's an expectation from this community and how we search for specimen-based data might be very different from some of the other communities from which we're linking data. So if I represent a publication-based community or a genetic resources community, or I'm a high school science teacher, or whatever other people we want to reach, thinking about how they prefer to access their data.
So if we're thinking a program that's going to run it, then who has to have that program to contribute? Does the contributors have to also have it installed? Do the people like the students that are using it, do they have to have it installed? Is there a license fee for that? I look at the way we're doing this now online, it's all open source almost. Once the institutions provide that data, it's available to everyone. So
following on what Libby just said, of this could be anybody doing the search. We had the idea of trying to do like a chat GPT type model where you kind of describe what you're looking for and saying, I want to find other things collected by the same person. And it responds with, this is what I think you're asking for. And here are some preliminary results. And then you can kind of iterate and refine based on that.
We talked about kind of re-envisioning monographs as kind of this place for like where a digital extended specimen could live. And like, since that's kind of a place we go to for getting all the information about a certain group of species or whatever level you're looking at already, kind of re-envisioning, I can't talk, what that looks like.
So something we didn't talk about is getting data into the system. So that kind of interface, which is also, I think there's competing ideas about data entry screens and or inputting a spreadsheet. And then there's the model of, here's my data, here's the API, have at it.
So just a question, because I'm curious, do you consider a question to be data? An example would be, I would like to see all the related records to this specimen. Is that data or is it some kind of ephemeral nothing?
But I guess it would be usage data, and it could guide refinement of the infrastructure, or yeah, it could also be used by governments in a positive way of seeing what are important questions, what do people care about, something like that.
There's a couple other hands up, but we're sort of seamlessly transitioning into our discussion, which is wonderful. But I would like to ask a couple things of the group. It would be great if you could snap a photo of both your bubble diagram and your user interface and drop them in the slides presentation. And that way we'll just have captured some of the brainstorming. And then we go to the next slide. And then just a reminder to put some things here on the idea map in the very next slide. Next one. Okay, so I'd like to re-invite our panelists to come up. And then also we didn't plan for a break because who needs more coffee or the bathroom? So if you do need any of that, feel free to step out for a moment. We'll just take a couple minutes to put your pictures in the Google slide and people can sort of reconfigure their seats, and then we'll just continue this discussion guided by a few other questions. Panelists, come on up. Make your way to the front. And, oh, Jorrit, I think everybody's actually here. They were just hidden. Yeah. You were too busy extending your specimen. And will you run the mic to?
Mike is, yeah.
Okay, the talks that you just heard in no way reflect the entirety of the work being done in this space. And so we're joined by several other people with relevant expertise and likely more than that even represented in this room. So I want to give them an opportunity to provide a little context from different slices of this conversation. And Conrad, let's begin with you, just while Jutta is sitting down.
Sorry, I missed the intro there.
Oh, just tell us who you are, what you do, and how you're operating in this space.
Okay, my name is Conrad Schauk. I'm team lead of NCBI taxonomy, which is taxonomy and name-based database that serves as the resolving taxonomy hub for the INSTC that includes EBI, ENA in Europe, and DDBJ in Japan, and then GenBank and other NCBI resources. So we are focused on the names themselves, providing the best accurate way that the names can be used. We also have a lot of informal names. So whenever somebody submits a sequence to NCBI and it's a new name that's new to the database, we look at it and we curate it, essentially. Another part of what we curate is type material that's attached to the names, and then trying to find sequence records that's attached to that type material. We also add publications, authorities, and various other data elements.
So representing the sequence link that many of you put as the data you're after for your extended specimen. And next, Jutta Buschbaum.
I'm a data architect since a month at the NHM in London. I'm also together with Libby, Andy Bentley, and Jyotsna Pandey, one of the organizers of the International Partners Group. And so this group is about facilitating the development and specifically the implementation of the digital extended specimen concept. And that across continents. So aligning our initiatives, informing each other, the different communities around the world about what we are doing. Yeah, and the important thing is that it is about people, our development. And actually there are over a dozen people here in this room who are part of the International Partners Group. And I would like to ask you to stand up if you're a member of the International Partners Group, so that we can actually put faces to this group. So we are actually having, thank you very much. So we are actually having 4 continents in this room who are represented in the Partners Group. And I would like to point out Alina, who is from Ecuador as a member of the group, Keping, who is from China in the group. Yeah, and then our asked Europeans and Americans. So yes, thank you very much.
And if you did not see Jutta's talk yesterday, on the conference wiki, there's a link to her presentation in which she talked about that working group. All right, Laura Russell from GBIF.
Hi, I recently had a title change, so it just happened this year. So I'm the training officer for community and capacity, which is actually what I was doing with that title. So it really reflects what I do now. But so I have responsibility for developing training curriculum, organizing events for both the data mobilization side of our community and the data use side of our community. So within this realm, once at GBIF, we get to a point of a new data model, things like this, where does extended specimen go? It's very important to me that I can develop the training materials, which we also do in conjunction with experts in our community, so that we can get adoption for all of these new things and ensure that people can share their data, and on the other side of it, that the users can use the data. Previously, I worked with VertNet for 9 years, so I helped people publish their data. So I've worked a lot with the collections community on that side of things.
That's great. And then I'd like to give Nikki a chance to talk about something that's relevant, and then I'll open it up to anybody else in the room who would like to add a flavor, either from your background and what you do in this space, or complications or desires or anything like that.
Yeah, thanks very much. So I was just a plug for a bit of work that I've been doing with.
You want to stand up just so people in the back could see who's talking? Yeah, great.
Yeah, thanks for giving me the opportunity to do this. So one of the tasks that we were given today about linking up data together is very similar to some work I've been doing called Echinopsis, which is a desktop system that allows you to work with semi-structured data, so work with images or text represented in Markdown and link across to data resources. So to GBIF, work for a botanical organization, so botany is very much my focus. I was a bit upset that we picked the fish, or I could have shown it to you. So yeah, you can grab specimens from GBIF, images from GBIF, literature from Crossref, names and types from IPNI, people from Bionomia, institutes from GR Cycle, and then link all those data together. We're trying to build this as a place where people can work today, but we will eventually plug in AI models that will do some of the linking and clustering of specimens and images as they become available. So I think I've got a slot to talk about it like last thing today, but it seemed very relevant for what we're doing here.
Your talk on this is today. Great.
Thank you.
Is there anybody else in the room who would like to provide any perspective on the digital extended specimen? Gotcha.
Hi, I kind of want to ask a clarifying question, actually. And it's a little bit about, it's a sort of a guided clarifying question about how data flows through the system as it is. And I was just thinking about it when Tim was talking, when Jorrit was talking, and it's about how data is accessed and where it comes from. And so I guess the question is that, so data sources come from museums and other places and data sets that are published, and that is this true that like GBIF and Global Biotic Interactions are indexing those data sets, but in just different ways? Right. So in some ways, I mean, I guess in some ways it's just repackaging kind of the same data sets in different perspectives.
Okay, we've launched right into the questions.
Oh, is that a question? Oh, sorry.
It is. That's great. That's what we're here for, to talk about our questions.
Yeah, you can. I see it as repackaging of the same data or interpreting of the same data, and whether they're the same is, depends on the when you look at it, how you look at it, how you save what you've looked at it, or when and record when you looked at it. So Globi might have indexed something yesterday. GBIF might have done it 5 minutes ago. They might be different. And if you keep track of exactly what you've looked at, and you can see exactly what was interpreted. So that's one of the things that I think is kind of important to do systematically, just like it is very important to keep track of your field notes or your specimen. We need to keep track of the digital resources we use in our respective systems. That's my take on it.
Does anybody else want to add to that? And if anybody would like to add to it, it doesn't have to be people at the front of the room. We have a powerhouse room right now.
I would add that the one thing we've really focused on over the last, 12 years with having institutions publish their data for sharing was that you publish it once, and then all of the different aggregators can use that same data in their portals. So it's not necessarily on the publisher of the data to get their data into all the portals.
This actually brings up an idea that has been talked about in many different contexts in this community, which is that how ought the data flow into these various programs. So if everybody, one model, which Andy Bentley couldn't be here today, but one model that he talks about all the time is that everybody should publish to one thing. And then from there, it goes to the downstream aggregators or the data sets or the extended versions of itself. But there's one spot where that data lives or is pushed by the individual collections. And then from there, it goes everywhere. And that prevents the duplication problem where the record in my database is different from the record in GBIF, which is different from the record in all of these other things. So good, some hands went up.
Yes. So I'm thinking more, and maybe Andy thinks in a similar way, but so I'm thinking more about a distributed system, like the internet. So it's not that we publish the data to one repository, but we have one node or one institution like the internet that has that, like in the internet, we have a central organization who organizes all the domains. And we would need to have one central node that stores all the, so I'm not sure if I expressed this right, but so we have one central repository that links then out to where do I find all the information on my fish. But the information itself might be distributed around the world. That's kind of more how I think about it.
That's your hand up first, I think. Oh, really? Yeah, so I want to say that just like the cassette tape, remember the cassette tape? It's still around. You can still use it. There's still, shops where you can buy it. Like think of the internet as the cassette tape of 20 years from now, right? Because it will be. Internet is just a cool little toy that was invented 40, 50 years ago. It's got popular when Hotmail came around and we could start sending emails in like HTML pages, right? But it's just one of the many ways that we can exchange information with. And what I'm concerned about is that we orient all our thinking and architecture around URLs and other internet related things, because at the heart of it is really digital and analog data. And I could put digital data in my notebook, I can write down 10111, right? It's going to take a little bit longer. I can put it on a hard disk, send it by UPS. So I think whatever we come up with in our community would be great if there would be like nice internet related resolving technologies, but I would still like to have a copy of my data under my pillow and want to be able to verify whether that copy is the same one that is also stored at the Library of Congress or the Internet Archive or Zenodo or whatever, right? That's what I wanted to add.
I just wanted to say real quick, but that, I think one of the things that GBIF does really well is it creates a registration, publication registration for those available data sets, if you know where to look for them. And then from there is just a list of all the available data sets that people can do all kinds of other things with. And so that's one of the things that I think is sort of missing, perhaps, is like a really transparently available list of where those data sets live.
So somebody mentioned a while ago about the getting data back to the local collections. And I know that's a hot topic for a lot of folks. I think it's a non-starter, frankly, for a system like this. If you begin, let's take GBIF as an example. If GBIF had the responsibility of taking all of the data and any changes that were made to it and sending it back to 1,600 collections in the US, that would stop the whole process. I mean, I think if there's a way to get that data, you were just saying you want to see your data and how it is expressed, I think that's the solution. I think the round tripping idea is, I mean, it's going to be possible at some point, I'm sure. I don't think it's possible now without a lot of wasted development time that won't satisfy people and will probably make them angry, because you'll be sending data back and they say, but that's not my data anymore because you changed it. So, I mean, I think it's got to be in some location like you're talking about, Jorrit, but it doesn't have to be sent back to the collection. You know, the desktop that I'm working off of, you know, like one collections manager told me when I mentioned this, she said, don't send that stuff to me. I don't know what to do with it. I mean, I don't have time to deal with all that flow of data coming back to me that came from my system. And I agree with that 100%. So I think we have to be careful in that regard. I know there are ways to solve that problem with distributed systems, but it's still a big problem that may not provide us a whole lot of benefit as the collection community if we still have a place to go to get those data and how they've been interpreted.
Is there anybody who would like to respond to that point right there? Because that has been a hot topic. You know, where do the data live? Does it come back to the home database or not? And Gil's making the argument that it should not. It would be a game changer. Yeah.
I guess all I'd want to add is that I think we should at least give people who manage the home of the specimens data in its original form the opportunity to accession that data and to add it to the record. Whether or not they choose to is another thing, but at least giving them the opportunity.
So I may be missing something here, but I'm, you know, as I'm thinking about this, we've got, you know, what Andy always calls the idea of getting stuff out born digitally. But the truth of the matter is, once it gets out there, it may be improved upon. And I mean, the classic example is GenBank. And is it the responsibility of the collection guild in that sense to keep track of what goes out and what gets published in terms of DNA data? Or is it really not possible in a digital world to repatriate that information to the collections? And I think the value of it is not really necessarily to the collections, but it's to the community with respect to things like, what if that happens to be misidentified? And, you know, again, that circle then can potentially feed back into the broader picture of making sure those data are correctly identified. And I apologize if I'm misunderstanding this.
fine, but I don't think it's a matter of possibility. I think there are possibilities, but I'm not sure they provide us the benefit that we might want from those. And so if data are published and then somebody annotates those data, just like we do now in collections all over the world, then okay, I think going to get those data and bring them back to your own collection if you choose is fine. I just don't see how it's responsibility of a digital extended specimen system to make sure that everybody gets the data back for their system. I mean, there's so many complications involved in that. Not to say it's not possible, just so complicated, and I'm not sure the benefit really satisfies the effort.
Anthony, I think you had your hand up. You still have a comment?
It was probably to an earlier thing.
Nikki, did you have your hand up? Yeah.
Thanks. It was really in response to what Katya said a couple of comments ago. That was how GBIF does a really good job of managing the registry type data by identifying publishers and data sets. And probably what we need to do here is like jump to the next level of granularity. I mean, it's great that we can see where data sets have been have contributed records to things, data downloads that have been cited, but we can't do that on the data level, on the specimen level. And that's probably the jump that we need to make.
Yeah, so one thing, just responding to you, Nikki, I think we are already doing this, because I actually showed you an example of linking from Arctos to Globi, right? That basically gives Arctos users the ability to see how messed up the interpretation of that record is on Globi, right? And I can point to many numerous examples how this led to this linking, this very shallow linking, because it's just a link to a query, that's a dynamic query that drops you into Globi, right? For that specific specimen, that led to all sorts of cool discussions about why did you call it this way, or why do I see a frog instead of a bird, right? And that led to all sorts of cool discussions, interactions, introductions for collaborators to meet. So the linking is happening. It's just maybe in communities that you don't, or that maybe we're talking about something else. And then to respond to Gil, I can see how it's different to merge data back into your collection. But I would see it more like annotating, putting like little post-it notes on your data, basically saying, Jorrit said this about your data, and then the collection people can say, well, we don't care about him because he doesn't know the subject, right? But if you want, you can have that interpretation show up. So that's already happening. Yeah, and that's already happening. I've showed some examples. Other examples are out there as a result of collaborations between people and institutions.
Yeah, so what I think this round tripping is not a must, but I see it as an option, as a possibility. So how I imagine community curation or annotation is that there is either one central node or a network of nodes that provide, or infrastructures that provide community curation and annotation functionality. And there only you would get like a record of what people contributed to your data or your specimen. So all the nodes, it's basically a list of what people remarked to with regard to your specimen or a specific link or so. And then if you are interested in what is happening out there in the world, what people are talking about your specimen, you could set a query and say, okay, if somebody remarks onto my specimen, I would like to get an email or an information that something happened there, and I can then look at that. So as a collection manager, I can look at that and then decide, do I want to actually like join this discussion? Do I just want to accept it or reject it? So it's completely, you are at liberty to how you approach this.
I'd like to switch gears a little bit and provide a use case that causes me constant frustration in terms of the extended specimen, which I'll put Conrad on the spot a little bit. But maybe you've had this experience. Somebody says, can you please send me these specimens? I'd like to sample them for DNA. And maybe they put an annotation label on them when they come back. But you know they got sampled, but so they got sequenced, and then it just disappears into the ether. And I think as a community, the most basic extended specimen is here's the specimen and here's its sequence data. That is like a link everybody wants all the time. And it was a link that was absolutely necessary during COVID. And I think the biodiversity community didn't respond very well to that because we didn't have the tools to make those linkages very quickly. And so most people now go through the painstaking task of hand linking their specimen data with the sequence data in GenBank. And then the other layer on top of that is that then an expert comes and looks at your specimen and they say, oh, the name is actually this. And then there's no connection for changing the name in GenBank because you're not an author of that sequence. And so that's sort of a constant source of frustration. And I wonder if you could speak to that a little bit.
Yeah, I don't really have a good answer for you at the moment. Essentially, what you need is an institutional submitter together with the individual submitter, because then it would allow you as an institutional submitter to make a change. So current system, you know, the archive part of the database is that the submitters owns, if it's 50 submitters, any of those submitters can make the change. We also have other partners in our INSTC collaboration that treats things slightly differently than we do. So if it came in through one of the other portals, you know, don't want to go into too deep into the weeds here, but if it came through EBI, for instance, there might be different rules at play there. I think that the solution is to find a way to make you as a biorepository a part of the submission as an institutional submitter. And I'm trying to have those discussions. You know, it's a bit out of my specific group, but that would be part of it, I think. And also to ensure that your specimens are correctly annotated with your, you know, correct institutional name would be another important part. And we know that submissions are incomplete, et cetera, et cetera.
Yeah, there's definitely if we could just change people's behavior solution, you know, if people, when they submitted those sequences, they had a voucher of what they sequenced. And then if they put the catalog number of what they sequenced in the form, there is a spot for it. And, but it's not required. And so, you know, that's one solution. And then I envision some kind of user interface like we brainstormed here, where maybe it's another program that can search GenBank or those off, and, you know, usually there's a third-party connection, which is a PDF of the publication in the appendix, which lists all the specimens that were sampled, you know, and so that is a three-pronged problem right there. Sharif.
Yeah, I just want to just quickly say that in Europe, we are kind of discussing these issues with the ENA and also with BOLD. So one of the things we're trying to do right now is do some metadata mapping. So BOLD, the metadata elements that BOLD has is slightly different than the ENA metadata, but there are common Darwin core terms. And one of the things will be there is the voucher specimen ID or the specimen ID. And ideally in the future, what we will propose is that that could become the DISCO specimen ID. So you have the DOI there and should be enforced during submission. And then from that DOI, you can get all the metadata, institutional ID needed, and of course, the submitter, things like that. But still, it's a quite a few years until we get there.
So I can't help but think that maybe taking a step back and starting to think about like the destructive sampling agreements that we put together as before even sending specimens out, and maybe the requirement to share to gene bank and to cite specimens should be included in some of those destructive sampling things, so that we have to basically, if they don't do it, then you can point back to say, can you finish your portion of the agreement? And that might help to kind of push this to actually happen.
Yeah, to follow on to the DNA conversation as well as the annotations, something that used to keep me up at night when I was working on barcoding is that a lot of identifications that end up in BOLD and GenBank are determined based on somebody sequencing something unknown, blasting it, taking the first hit there, and then that becomes the organism name that gets published, which then becomes a source of truth. And that perpetuates, and that really bothered me. But then I moved away from the barcoding group and I got better sleep. But yeah, I think that's still an issue.
So I would like to, you want to go first?
Yeah. I guess it's sort of a kind of a sub-tangential question is, for those of you who are in charge of providing loans and answering loan queries, what I guess systems or consequences are in place for those who are poorly performing on the other side of the loans? And should that information not be linked in some way to that person, to them as a collector or as a provider? I mean, it seems a little absurd, but I mean, how many of us have used Uber or Lyft and we see that you as the rider also have a review score that the driver can view, and that is information that they have a right to know?
So sounds like you're looking for a service that allows you to review your data set, right? And find all the DNA sequences known for your specimen. And I'd say, let's build it. Let's do it. As long as you give me a
Yes,
coffee
please.
and a cookie.
Cookies and coffee.
No,
It's so
but,
easy.
like,
God, I didn't know.
No, but I'm serious, because we have the capacity to build very specific, focused little robots that can do this very quickly. As long as you can give the examples, ask very specific questions, then we can build it. That's really my limitation. I don't know to come up with the right examples and to have reviewers that basically are able to look at the automated reviews saying, hey, this specimen should have a sequence and I don't see it. So you want to do it?
Yep.
And sorry, I think part of the problem is that the specimen reference doesn't end up into GenBank. So even if you had something to crawl to look for them, a lot of the people that are submitting their data aren't actually including anything saying that it came from X museum.
Yes, and I would
At least we could do it for those that do.
Right.
And many times you don't have the URLs anyway for those that are cited.
So what I'm hearing over and over is that their statements make, I think, or this is reliable, and you can measure these things. You can make, you can do reviews of data, and you can have very specific results on that. So I think we should just do the work, see how far we get, and then we can say, well, 20% of sequences of Jen's data sets were appropriately annotated and the rest was not. And then we can iterate and improve and get more clarity on what we need.
Conrad, Tim, and then Teresa.
Yeah, just to put some numbers on it from a taxonomic perspective, I just did a quick search this morning for fungi. This is working. So fungal species that were published in 2021, 25% of them are not in GenBank. So I would expect most of us to have sequences, but they've just not been added correctly. So that's a challenge to find those and have the names attached to the sequences, because they're probably in there under an informal name, maybe for specimen or maybe not.
Thank you. We were talking about how do you link GenBank data to the specimen data, and Sharif's pointed out in Europe we're trying to work with the European side of INSDC to improve the submission protocols. The clustering algorithm that I introduced is actually doing what you just talked about, where it is indexing the data that's published out of the INSDC, GenBank databases, all the specimen data, and trying to find links between them. The metadata is hugely variable in GenBank. There's a mix of people's names, there's codes, there's locations it was collected. It's semi-structured data. What we try and do with the clustering stuff is detect links using a variety of those dimensions, those measures. It's not just like catalog numbers and codes. I've spent quite a bit of time trying to improve that. I don't think it's going to get that much better. A lot of this stuff has got no information at all about the sample that it came from, and we're never going to be able to link those records. I think Sharif's probably correct that we need to focus on improvements for the future rather than focusing too much effort on trying to link what's already in there, because I think the data's already lost.
So first of all, it sounds like GenBank needs annotations so that I could just go say, hey, this thing belongs with my record, not change the record, but say, this is mine. I have the specimen. There are people who have made crawlers for GenBank to go look up identifiers and then come tell you, hey, I think this came from your specimen. So that's a thing. But really, all of this relies on good identifiers. And if what people take from you is catalog number 1, 2, 3, 4, 5, and you expect to get back some good information, it's probably not going to happen. And I know a lot of our collections, we don't have much more than maybe a code and a number. And there's no guarantee that that's unique. And there's no guarantee it's going to be written in the way you think it's going to be written, if it's written at all. So half of this is a social problem that, people need to be aware that they should be citing things, but they also need a way to cite them that means it's unique in the world, which is big right now, so that you can figure out these linkages. And it's not just you, it's anybody who wants to make use of this data out in the future. To some degree, I would argue that you shouldn't have to make that link. If you're a collection manager, you don't care. You've put an ID on your thing. Other people should be using it. And over time in this distributed network, they should be able to link up that GenBank sequence with this specimen.
Yeah. Oh, sorry. I actually just wanted to add kind of a connect two thoughts that we had earlier. I think that you mentioned about asking is a question data. And I think if you think about loans, that's a question, right? You're asking a question, and you're trying to get the specimen because the specimen is your question. So I actually see no reason why something like a loan request shouldn't be linked to your org ID. Because that's providing data on a question that you've already asked. It links you to the person and it shows a request that you made at some point. It just, I think, it simplifies a lot of that system.
then you get 1 star. 1 star on the loan return. We have just a few more minutes and then it's going to be lunchtime. We have a lot of people in the room who are at the table when important decisions about the extended specimen development are made. And so are there any final thoughts or things that you would like to convey to the panel, to the room? And we'll go Yuta, Katja, and Anthony.
So one thing, we are talking about concrete functionality and projects. And I attended Spinach last week, and there were some really great examples about local extended specimen infrastructures. For Yale University had this really great presentation about their extended specimen implementation. Indiana University had one. And then there's these specialist networks who do it too, like you are doing. So we have all these islands of really, really great examples of use cases of the extended specimen. So now I am this kind of like really abstract person, and I'm thinking about how can we actually connect all these islands? We would need a matrix that actually allows these islands to talk to each other and be connected. So I'm thinking about we would actually need kind of like a harmonization layer, like something that maps from Yale to Indiana to Gobi. And that I think the one difficulty here is that this is a layer nobody of you is, or maybe no, many of you or many of the users won't see, because you shouldn't. You should not need to think about this kind of like really high level abstract backbone. It should function. And you should be able to build your crawlers and your clustering algorithms and everything, and it should work. But to have this function, we need this kind of like matrix that connects everybody. And I think that is something that maybe we should think about. So.
One of the problems I've had working on a checklist is data access. And I think that there should be some thought of maybe pressuring politicians to change some of the laws like in data access, because it doesn't make any sense to have a monograph that can only be accessed through a paywall, even though it's published 20 years ago. I mean, whoever published it has returned their money, 100 times. And also how to finance small operations to get their stuff digitized. There's one specimen I had that the only voucher I have for Colombia is in a small herbarium in Italy. And I would like to have access to that information, without having to send a letter to the herbarium.
I guess one just parting thought was as a director of a bio collection at a research university, one of the things that I'm starting to get more and more requests for are supporting data that actually have no specimens. So there's not actually a physical object anymore. And this largely comes from a community ecology folks where they, and with virology and bacteria related things or environmental DNA, where they're sequencing and they're making some assumptions, but based on those DNA sequences, there is information about occurrence and other things that fit, I know well, into sort of GBIF models, but the specimen is not there. And I just wanted to, just remind folks there may not actually be a specimen within the specimen network.
The extended nothing. Okay, everyone, thank you so much for your participation today. This has been a great section. Let's again thank our speakers. And and it is now 12 o'clock and the plenary begins again at 1. So go get yourself some lunch and we'll see everybody back in the main auditorium at 1 o'clock.